Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, once again, welcome. We are today having a session about Unit 3, chromosomes again, like we had seen previously. And within this session, I want us to pay much attention to a few small details, a little bit interesting because we are nearing towards the end of the lesson. Remember, we just have the word continued, meaning that it's an extension of the previous lesson we had. Now, it is indicating that we are on the third objective in which we will look at the role of telomeres. Please, I want you to pay attention and focus much and see how interesting this is going to roll out to be. Now, when we look at the telomeres themselves as structures, the question comes in, taking us back to what we have been looking at before as a chromosome. If we can get back to what we had previously, we are looking at the telomeres as components of a chromosome, like we said. If I can use this section here, where we said that they are some small structures that are resembling the caps or locks which we have on laces of shoes. And we have here a very good example I've brought forward for you to be able to look at what we are talking about. This would represent my chromosome, and you have a knot in the middle, then you have this section, and this, and then that. Now, there is something I've tried to change a little bit, as you can see, that this section is kind of frayed apart, and then this looks intact. The same applies to this slightly, and this is really looking intact. So we would compare, therefore, this and that with almost intact laces for your shoes. Then this one kind of fraying apart because the cover, the other plastic thing we talked about, is off. So taking this and dragging it here, I think I've made a simple kind of representation by a simple model. This is just a thread I tried to design, so you can as well make your own at home. Now, we say that these telomeres are located on these ends, and here we say, one, located at the ends of the chromosomes, like we have indicated, we have mentioned here. Now, the question is, what should they be for? So going back previously when we were discussing about aging laces for your shoes, how it becomes very hard for you to put them on and you have to start struggling, fix them inside your shoes and the holes on the shoes to be able to tighten your shoe, and it doesn't work very well. The same applies to chromosomes. So, literally speaking, one would say, you look at a shoelace, think about a chromosome, and there you have it. Now, they are located at the ends of the chromosome, and we need to start thinking like we are just having a structure. I got a small emoji here for you, which has make you think that one, you have the lace. Sorry, I need to change this to C, a shoelace. And we're also taking us to the wearing away of these laces, as we have just talked about. And one, looking at how the cover, the plastic top on a lace gets off and then starts aging and you throw it away, look for another one, is in the same way our telomeres have to maintain the structure, keep it intact, so your shoe can be used for much longer. Two, your lace will always look new. Now, so it maintains and stabilizes the chromosomes. So it means, therefore, we have the sealing, like keep tightly closed. Two, we also close up, keep the end tight. So we would therefore say this is not tight and this is tight. That's what we're trying to look at here. But also, the reason should be, if we go back here, I want you to look here. If this inside is our DNA, which is linear, so it has a linear arrangement, okay? At the very end, we must have a seal. Like normal length, it's only here and here. So they'll keep the ends sealed, and so during the time when the cells are dividing, when cells become many, they keep multiplying, the interesting part is that these have to make themselves more of the copies. Uncoil, stretch out, make a copy, load the copies into new cells, then we have more cells coming up. It therefore means that the same seals, the same ends we're talking about here, are very important when cells are dividing. So let's get here and check. The usual expected components of life, this is where it gets interesting, like I said, pay much attention. They shorten with time up to when we have a critical length where the cell can't divide anymore. Now the question is, if cells do not divide, to have new copies, have more cells building up in the body, am I going to remain this size? Will I start shrinking? Do I get old? Which comes into this interesting part of our life as ordinary people, where we do not know actually being old and aging, the connection or the relationship, or if there is any difference between the two. Now, check this out. If your laces get damaged, you have a problem. It becomes very hard to put your shoes on. The laces start wearing away as we are looking at, they fray apart, and they may break down. Now the question is it in any way connected to aging? How do we get old? Is it before we are born? Or right away from day one and 1 year of your birthday, and then onwards. Which is so interesting, where now one has to ask himself, oh, my dad hardly thinks about celebrating a birthday. Why? Does he have a fear? At your age, year 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, you always wait for that cake and you love it. But the question is, are you heading this way? And what happens? Do you ever want to remain young forever? That's a very interesting question. Do people enjoy getting old? Yeah, you want to show some bit of maturity? Yeah, that's wonderful. But how does the word old and age, if you link the two words, what is the connection? How do you find it very easy for someone to refer to you as aging, at the same time someone referring to you as someone who is maturing? Now that's the very other interesting part of it. So, we have always thought that some of these diseases like cancer, cancer is for old people. Cancer is for people who have aged. But do we have cases of young children, teenagers, 30 to 40-year-old people also having some forms of cancer? Where do they come from? Could it be something wrong happening within these chromosomes? Now, when we look at the telomeres, and a link to any of these, they're saying that they have a repeating sequence of bases made by the RNA, and they contain enzyme. Now, let's go back to somewhere we were looking at the processes of reactions in the body, that they have to run at certain speeds. Then there must be having some natural controllers in the body to be able to give us what we need. So the reaction does not go so slowly, neither does it go so fast that the body cannot control. And this takes us to enzymes, which you must have seen in your grade 10. Now, here we have, if these are the telomeres, then the enzyme involved in all these processes, therefore, all we need to do is extract this word, bring it up here, and name the enzyme. So we have telomerase as an enzyme. ASE. Remember, that's the rule. Get a structure, a reaction, and then add ASE, and there you go. Now, what is so interesting also that we need to note is that under normal conditions, the enzymes are suppressed. They don't work. They are inactive in normal cells. But when you have a cancerous cell, a cell that has gone out of the way, performs different activities compared to the others, does something in a wrong way, overconsumes the materials, overproduces the wastes, divides much faster than the others. I mean, so wrong that it cannot keep in one region where it was actually meant to be. Now, when a cell is cancerous, the enzyme stays active, and therefore, hey, this takes us back to the expectation. So I want you to find a connection between this point and the expected in our bodies. Look at the connection and think about it. I think we are good to go now. Under normal circumstances, telomeres would shorten, so the chromosomes get shorter, to a point where you cannot separate these chromosomes to have more copies within the cells. But when the cell turns into a cancerous one, do wrong things, have poor cell membrane, ooze out substances, like as in do the wrong thing, like the other bad animal among the others. The enzyme stays active, the length stays, and so much is happening. Now, the interesting part here is, what would happen? How would we make sure that that does not take place? Have we been in position to check how important these telomeres are? That without them, then something might happen. Without them, a lot of things can happen within our cells. Which takes us to some of the activities we've been looking at. But before we go to this, I want us to remind ourselves of a few things we looked at previously about the structure of the chromosome, which was our core component. The overall picture of the topic, of the lesson, should be if we are in position to understand the structure of the chromosome. So in terms of structure, we're looking at the components, maybe the design, what other accessories are around that we need to include if we are to look at the structure. It's like someone saying, how do you describe your sister? How do you describe your brother? And so he says, he's this tall, and these meters, a few inches, and puts on this type of clothes, and likes high-heeled shoes, or all the time, you're always having a wristwatch. Now, that's a simple description of a structure. You have the following, and they are arranged this way. That's what we look at. Someone would say, maybe our chromosome is just this. So now you have to think about, if I was to mark this learner, can I give this person a 0 just because she has said a chromosome is this? But I would think that this person is not wrong because he's giving you an impression of the design, which is very correct. Now, when we get to the activity here, I want us to go slowly here, and we look at 1. When we look at the activity that has been provided here, so we are doing this together. So it's a do now. So I need you to focus. Look at it very carefully. Now I want you to pay much attention to some of these things that make us go wrong even when we know what is expected. 1. Language. 2, a command word. 3, a term. 4, the stake. You can't go into a deal not knowing how much you are going to get out of it. I won't gamble unless I know the risk. That's what I'm trying to say. 2, centromeres contain no genes. What? It's just one word. It's just asking. Just straight, precise, simple, and very short. Main function. It's not S. It's only function. And then the third component, they are also asking us what we know as a nucleosome. Now, this is where I get you. The previous lesson, we talked about these things. How much of it can you remember? So on a count of 5 to 0, I expect that you have got close to the screen and taken a caption of this. If you have a phone, you can get close and snapshot it. So since you do not have the time to actually do all the writing, so with a phone, just get close. Those who have the ability to record, you can have it, and then you can review the lesson after. And then you're able to do some of the things. Now, to make a little bit of the work much easier, if I put an arrow here, then we are here. Now the question comes in, how far do I go with the question? Do I just say, describe the structure of a chromosome? Teacher, I'm going to put an X and then that's it, I'm done. We have to ask ourselves, if I just draw X and this, and I'm done, do I get the 3 marks? Or they're just going to say, well, based on that drawing, yeah, you have 1 out of 3, and there you go. They say, you come back complaining, you see, teacher, now. Let's focus and see, if we were to distribute the 3 marks across the whole question, where is the greatest stake? Description this is a command word. So I would say in Kinyarwanda, ni uruzingi. That is where the score is. 2, I need to look at the structure. So I look at the part that if I'm to describe 1, let me think about the components. Can I mention them? So here we go. 1, you have the centromere. 2, you have the chromatide. And so you have this, and then within the chromatide, we are having our helix, our strand coiled on another one to form our DNA. But then in the middle, you have that joint, that seal that puts them together. So it means that in responding to the question, I have to think about, one, components, arrangement, design, as in the structure. Is it X-shaped with so and so and so, which takes us to this arrow. Take a look at it very carefully and see. From here to structure and then there. It means, therefore, that in addition to the word statement, there is an option to draw. Remember, you need skills to draw. At some point, you need to learn how to have the pencil roll on paper precisely, in which one would look at your outline of the picture. 2, are you labeling it very well? And 3, what is the proportionality? Are you going to draw this structure and it fills the whole book? When it comes to the second question, we come here. A region of the DNA that is responsible for movement of the replicated chromosomes into 2 daughter cells during mitosis and meiosis. These 2 words, would be an equivalent of saying during nuclear division. Remember what we said, that this makes copies of itself several times. And that's what we talk as mitosis or meiosis, that we look at as nuclear division. Not cell division, but nuclear division. The last part, 2 marks, would come here. A structural unit. Something so interesting. You do the math, and you have the word unit as the smallest measurement. So if you are looking at the whole structure of the DNA or a chromosome, there is always the smallest part. If we do sets, if we do units, if you can go straight to your grade 10 about biological molecules, you have proteins, they have amino acids, you have carbohydrates, they have sugars, as glucose as an example. So those are the smallest components possible. The same applies to this, that a structure unit of a eukaryotic chromosome consisting of a length of DNA coiled around a core of the histones, which takes us back to the previous lesson about that structure, in which we were referring to it as a yo-yo, if you can remember. The rounded structure which kids play with a string in, and just that component is the unit we are looking at here. And then we have extension. This is now your take-home activity. Let's go through it together very carefully if you get a close look at it. I won't be in position to read the whole of it, but try to get close and see what is contained there. The telomeres may be lengthened by introducing a gene for an enzyme called telomerase reverse transcriptase, HTRT, causing cells to produce active telomerase that repairs telomeres. So it's a component of science. It needs so much extensive reading, you need to research and understand much more. So they are saying, we need to give a suggestion pretty interesting. Another command word, when we say suggest, it means that any answer provided, as long as it falls within the boundaries of the concept, is acceptable. Now, if it's acceptable, the question is, how far am I going to enter and approach in depth, if needed, or support my argument about the question? And it says, suggest how the techniques for manipulating telomerase or telomere length might be used to treat age-related diseases and cancers, which takes us back here. How do we get old? When do we get some of the diseases? Where are these cases more rampant? Are they random? Is there a correlation? Is there a link between aging and some diseases so that we connect them? 2, what risks might be associated with deliberately? That's an interesting part. Deliberately doing what? Changing the length of telomeres in body cells. Now, which takes us to 2 important things I want to mention here. 1, I know you like movies. How many of you have ever watched Captain America? I don't know how they pronounce it in French, but at least you should be able to have watched that movie. If you've never watched it, you're really missing something. I remember there is this pilot who keeps up in air and then has something like a fiancé or wife, I don't know. I don't remember because I watched a long time ago. And this lady is aging, and the captain, who is the pilot, is actually still super perfect. Now, the question is, when you're watching the movie, what feeling do you get when you look at Captain America, looking at the former friend as a lady, lying in bed, too old? Same age, and one is just young, and the other is too old. The same applies to this question. Now, there is so much we need to look at and think about. When we look at telomeres, we connect them to how long we can live, longevity. Now, here are a few things. 1, any change in telomeres will affect how stable the chromosomes are. 2, they also affect how cells get older. 3, senescence, reaching that point when you feel that you are 50 years and you're same as 60. Like there's no difference. Time is passing, years are going, and you still remain the same age. 3, perhaps some forms of cancer. Now, I want you to do more extension and copy these. You must have taken captures, clips of them. These 3 links take you to extended discussion of how much you can know about aging, disease, and the telomerase and other structures. So with that, I want to thank you so much for attending this lesson and paying much attention. But one more interesting thing I want you to look at is check these links very carefully and take snapshots of them. Just type in, whether it's a phone or any other component, you should be able to get into so much information about how we get older, why we get some of the diseases, and how we end up, if we were to make mistakes to keep young, apply certain cosmetics, we have a problem. Thank you so much for watching, and I want to say that you try to continue reading and more, do research. It gives you much more than we can provide.